this question here, the one way of eliminating is last week and the week prior to that, we were talking about a specific class of compounds that are not flavonoids. So, if you can recall what we had discussed earlier in the last week and the week before that, that might give a clue, right? Yeah, Swati. So we have just started discussing. This is the second question that we are discussing. So the question is select the non-phenolic compound from the options given below. Quercetin, eugenol, linalool, and mandroferin. And I was just giving a hint based on what we had discussed in the past 2 weeks on the different classes of compounds. One of that is mentioned here, which is the non-phenolic compound. So if you can recall what was discussed in the past 2 weeks, that might help you. In the next slide, anyways, I will be showing the structures, which will make it easier for us to identify the non-phenolic compounds.
Is it option C, linalool?
Yeah, that's that's something that we discussed in the past 2 weeks, right? Yeah, and we'll look at the structures as well. So this is quercetin, this is eugenol, and this is mandroferin. While these two are normal flavonoids, this is a glycoside. It has a sugar moiety attached to it, which will make it more soluble. And the phenols here would either correspond to this or this in quercetin. There's only one ring here, whereas here you have this is another phenol and this is another phenol that's linked by an oxygen here and a carbon here. Whereas linalool, do you remember to which class of plant-based compounds linalool belongs to?
Is it terpenoids?
Yeah, it's an alcohol. As you see here, it has an OH. It belongs to the terpene alcohol family. Correct. Right. Uh, Maybe I'll just go through the first question. You were not here for that question. So the first question with which we started the class was select the correct pair of primary metabolites that serve as precursors for the shikimate pathway. Option A, erythrophore phosphate and phosphoenol pyruvate. Option B, glyceraldehyde 3-phosphate and phosphoenol pyruvate. Option C, ribulose 5-phosphate and phosphoenol pyruvate. Option D, erythrose 4-phosphate and acetyl-CoA. You can take a couple of minutes, think about the question. Maybe we can start by discussing what the Shikimate pathway is, where it leads to, and then uh, do we get any specific class of compounds that are present in all living organisms from the Shikimate pathway. We'll just start discussing there. You can take a couple of minutes, think about it, and then uh, you can start discussing with that.
I think it's option A, Shikimate pathway, erythrose 4-phosphate and phosphophenol pyruvate
Okay,
will form the Shikimate and then
correspondent. Yeah, that's right. Do you know what is the okay? I'd asked about two questions. What is the class of compounds that is being produced by Shikimate pathway? Do you have any idea about that after correspondent?
It produces finally the amino acids phenylalanine and tyrosine.
Right. It produces the aromatic amino acids, phenylalanine, tyrosine, and does it produce tryptophan as well?
Produces tryptophan as well.
Right. And why are these essential? Any idea? What class of compounds do we get from these aromatic amino acids? Any idea?
The indole alkaloids we will get from tryptophan,
We'll get flavonoids from this.
flavonoids.
Flavonoids, yeah. So you have enzymes like okay. Although Shikimate pathway produces all these three different amino acids, for flavonoid biosynthesis, we have either phenylalanine ammonia lyase or tryptophan, tyrosine ammonia lyase. These two amino acids, when covalently attached to an amine group, they would further produce the flavonoids after a number of steps. Yeah, we'll be looking at that later. So, like you mentioned, uh, we get phosphoenol pyruvate from glycolysis and erythrose 4-phosphate from pentose phosphate pathway, which gives rise to deoxyarabinose heptylosonate 7-phosphate. This further gives rise to shikimate. Right. We went through this question. We'll go through the next question. The next is identify the precursor compounds that gives rise to chalcones. Comaryl CoA and 3 units of acetyl CoA, comaryl CoA and 3 units of malonyl CoA, comaryl CoA and 3 units of succinyl CoA, comaryl CoA and 3 units of cinnamoyl CoA. So, in the flavonoid biosynthesis pathway, we have before the formation of before the formation of flavonoids, we have chalcones. Chalcones are first synthesized and then they are cyclized to form flavonoids. So now, which of these is essential for giving rise to chalcones? And what is the enzyme that is involved in that process?
I think option D, comaryl CoA and 3 malonyl CoA, the enzyme resveratrol and chalcone synthase, it will form the chalcones.
What enzyme did you mention? Resveratrol.
Yeah,
That is a compound. That is a substrate. That is not an enzyme.
enzyme is chalcone synthase.
Chalcone synthase, is that the one that uh, takes in coumaryl CoA and 3x of malonyl CoA?
Yeah, it
Right.
forms the chalcones. I told that option B,
B.
it forms
Right.
the chalcones.
Ha. Right. Like you mentioned, we need paracoumaryl CoA and malonyl CoA, which under the action of chalcone synthase gives rise to chalcones. Now, what is interesting here is, here it is not fully cyclized. Next, you have chalcone isomerase that cyclizes this to form flavonoids. So, like you mentioned, it's paracoumaryl CoA and 3 molecules of malonyl CoA. Right. The next question is, identify the correct reaction catalyzed by the enzyme phenylalanine ammonia lyase. Four different options are given here: phenylalanine to caffeic acid, phenylalanine to coumaric acid, phenylalanine to vanillic acid, phenylalanine to tranexamic acid. So this enzyme called phenylalanine ammonia lyase. So to think about this question, you can think about first the structure of phenylalanine. Once you think about the structure of phenylalanine, think what an ammonia lyase would do, and then maybe that will help you get the structure of the final compound that we are looking for.
I think it is option D, phenylalanine to trans acid by the PAL enzyme, phenylalanine ammonia lyase.
So what is the chemical reaction that you think will be happening here? Just from the name of it, it says phenylalanine ammonia lyase. So phenylalanine is your substrate here. What does an ammonia lyase would do? Any idea?
Ammonia lyase means ammonia will lysis will occur.
Okay, so and ammonia, or in this case, this is an amino acid. It will not have ammonia. Instead, it will have an amine. It will have an NH2 group. So
NH2 lyase. Yeah.
yeah, so this, it would be removed off from phenylalanine. Is that what you're saying?
Yeah.
Right. Huh. So this is how the reaction looks like. Once it gets removed, you have a double bond here. And this is the reason why you have the name tranexamic acid and not uh, just cinnamic acid. It can adopt either of the two conformations here. Cis conformation where both the hydrogens face each other, and trans where they are opposite. So this is the predominant form when this enzyme PAL is being used. Right. Next question is, identify the general formula of stilbene. Option A, C6C1C6. Option B, C6C3C6. Option C, C6C2C6. And option D, C6C3C6 as a polymer having N units. Yeah, we can think about this question for a couple of minutes and then we'll look at the right answer.
think it's option C, sir. C6, C2, C6 stilbins.
Okay, that is still bean. Do you recognize any other uh, kind of molecule that we earlier discussed about based on the structure? Like we had looked at pathways. One option that you can eliminate is this polymer because we didn't look at polymers, rather, we looked at small molecules. So, option D can be eliminated. Any idea about C6C1 C6 or C6 C3 C6?
C6, C3, C6. You're asking that is, I think, flavonoids.
Okay, so yeah, you're right about that. This is a still bean where you have C3, C6, C2. Again, you have a trans configuration here. Whereas for flavonoids, you have these two attached by C1, C2, and C3. So C6, C3, C6 is flavonoids. That's what you had mentioned. Good. The next question is, what is the outcome of antisense expression of HCT gene in alpha alpha? That is Medicago sativa. Option A
Excuse me,
in
sir.
yeah, sure.
The previous, then which is the answer, sir? Then
The
C6.
answer is C6, C2, C6. So for still bean, we have a
Ah,
C6 and then a C2 and then a C6. No oxygen, no third carbon. Only
okay.
C6, C2, C6.
C2 and C6. Okay.
Yes. Whereas this is flavonoid, general skeleton structure of
Flavonoids.
flavonoids. Yeah.
Yes.
Huh. This is an interesting question. So here they ask the outcome of antisense expression of HCT gene in alpha alpha. That is Medicago sativa. And they give four options: increase in G lignin, increase in S lignin, increase in all the lignins, and increase in H lignin. So you can think about this question for a couple of minutes. You can start by mentioning what G lignin, S lignin, and H lignin are. Yeah, can you hear me?
Sir?
Yeah, Swati, can you hear me?
Yeah. For this one, is it increase in H lignin?
Yeah, we'll come to that, but before that, do you know the difference between G lignin, S lignin, and H lignin?
Uh, no, so I forgot. Please explain. No.
Okay, so lignin has different monomers. One is hydroxyphenyl, one is guanacid, the other is syringyl. These are the three different units. Now these are the different components of lignin. That's why you have G lignin, H lignin, and S lignin. Now coming back to the question, they ask about antisense expression of HCT gene. Do you know what this HCT gene is?
Hydroxycomeryl hydroxo hydroxycinnamoyl transferase gene.
Right. So what this HCT does is, in 4-comeryl-CoA, it takes this shikimate group and replaces the CoA group. So we get 4-comeryl shikimate. After this, after C3H acts on it to give caffeoyl shikimate, now again this HCT converts this shikimate to CoA. Okay, so this is the reaction that's happening. So hydroxycinnamoyl CoA shikimate, hydroxycinnamoyl transferase. So now this is the step that is happening. So uh, do you know what antisense expression is? Antisense expression. Do you know where is it used, or what is the mechanism behind that?
Uh, Mechanism, so I don't know. Paracomaryldehyde, then it forms the paracomaryl alcohol by antisense of the HCT, which increases the H lignin.
Okay, so antisense expression means, so you have a DNA which gives rise to another RNA in the process of transcription. This RNA is the one that gets translated to form an enzyme. Now, suppose you have an RNA that is introduced here that binds to your RNA that gives rise to your HCT gene. Basically, you have a complementary RNA that binds to the mRNA of the HCT gene. Then translation will not happen and this enzyme will not produce. So when you put an antigen, when you do antisense expression of HCT gene, the levels of HCT gene will get increased or decreased. Will the levels of HCT gene get increased or decreased when you do antisense expression?
You are asking
Yeah,
the levels of antisense expression of the HCT gene.
no, no.
It will
When
then.
we do an antisense expression for this
Hmm,
HCT gene or
okay.
any gene,
Uh,
what will be the levels of the enzyme? HCT enzyme, will it get increased or will it get decreased?
it will get decreased.
Yes, it will get decreased because you have an RNA that binds to the mRNA and translation is not happening. So it will get decreased, right? So if it gets decreased, then the enzymatic process will get reduced. This has been highlighted in a paper. So what they did is they did the same experiment and they observed the effect of this in the different batches that they have. Here, if you see, you have a few controls. So what you need to specifically see is in control and in these cross sections. If I'll just read it from here. So what they mention is here these are the plants that are used for the study. B is the cross section of the fifth internode of the stems that are stained. And this purple or red coloration indicates the presence of S lignin. higher magnification of CK is controlled, and line 7A. Line is a transgenic expression line. So if you compare this CK control and 7A, it shows that there are xylems that are distorted compared to the normal vessels. That is what is shown by the arrow marks here. I will zoom it further. Unlike forming a well-rounded dome-like structure, the curvature is not good enough here, which is what they say by saying distorted xylems. So again, this indicates 1 minute. Mentioning this is the result of that study. So here they say HCT downregulation resulted in strongly reduced lignin content and changes in the lignin monomer composition with predominant deposition of 4-hydroxyphenyl units in lignin. So this 4-hydroxyphenyl is your H-lignin that is getting deposited, which is what they are saying here. Right. The next question is identify among the following the correct group of phenolic compounds within which podophylotoxin belongs. Option A lignin, option B stilbene, option C lignin, option D tannin. You can take a couple of minutes to think about this question and then um, um, try to answer this.
Is it lignan? Yeah. So is it lignan?
Okay.
Option E.
What is lignan? Any idea about that? Yeah. What
Uh,
is,
two
yeah,
mono two monolignon joins and it forms the lignan.
what is monolignon?
Monolignals.
Lignol. Okay. Hmm. Okay. We here, what is the monolignol? Are you able to see that?
It's small actually. Yeah, I can make out.
Okay, so here what they mention is these lignans are found by coniferal alcohol. That's what they mention here, specifically for this podophylotoxin. Like you mentioned, it's a lignan-based natural product. Again, it arises from the Shikimate pathway. And one thing that they mention here is there is something called as an arylation that is happening, and this is the structure of podophylotoxin that you see here. Right. The next question is fill in the blanks. The following sequence sentence for the formation of ferulic acid from paracomaric acid, dash and dash are required at dash position. So you need to know about the structure of ferulic acid and the structure of paracomaric acid, from which by the action of two different enzymes, two different processes happen. If you just look at the structure of paracomaric acid and ferulic acid, it will appear like it's just a single change. But if you carefully look at it through the enzymatic perspective, you will know that two enzymes are required. So maybe for paracomaric acid structure, we can look at the previous structure that we have. So here, let's say instead of the CoA, you have an acid. That's paracomaric acid, like here. Hmm. Alright. So instead of the CoA, and you have an alcohol group, which makes the COOH instead of COS CoA. Right. So this is paracomaric acid. Now, going to the question, we want ferulic acid.
Then what is the question? Can you show once?
Yeah. So the question is, how would you convert paracomaric acid into ferulic acid? For the formation of ferulic acid from paracomaric acid, dash and dash are required at the dash position. I was showing you the structure of paracomaric acid. Now, if you know the structure of ferulic acid, which I will show in the next slide later, you should be able to tell what is the minimal set of chemical reactions required to convert paracomaric acid to ferulic acid.
This is a structure, no idea, sir.
Okay, so we have paracomaric acid here. Paracomaric acid doesn't get directly converted into ferulic acid. This is the structure of ferulic acid. The difference between paracomaric acid and ferulic acid is here you have an OCH3 in ferulic acid that you don't have in paracomaric acid. This is a O-methylation process, but this cannot happen in a single step. It has to undergo this hydroxylation reaction first, so that you have an OH group. Then you have an O-methyl transferase that would add a methyl group to the oxygen here. This is the reaction happening. So now, based on this, will you be able to choose the options here? What are all the two reactions that is happening?
Hydroxylation and O-methylation.
Yes, and that gives us two options, and meta and ortho. Are you aware of these positions? Meta and ortho position. There's one more called para.
No,
This
no.
is, oh, okay. So in chemistry, based on a particular functional group, we decide the name or the position of other functional groups. For example, this OH and this group are opposite to each other on this benzene ring, right? So this is called as para position. This OH is in para position to this, right? So now that is why this is called as paracomaric acid. So if it is next, so for this particular carbon, if you have substitutions on either this or this, the immediate next carbons, it's called ortho. The second immediate is para, or second immediate is meta, and the final one is para. Okay. So now they are asking what is the position of this OCH3? Is it ortho or meta or para?
Meta.
Meta with respect to this. Yes. Ortho, meta, and para. That's the right answer. You'll have to choose it based on this moiety. Correct. The next question is the core phenylpropanoid pathway involves action of three major enzymes. These are PAL C4H CAD, PAL 4Cl HMGR, PAL C4H 4Cl, PAL C4H HMGR. You can take a couple of minutes to think about this. Uh, this pathway begins with either tyrosine or phenylalanine. So PAL is by default the starting enzyme, and that is present in all the four options. From PAL, what do we get? You can think about that, and then maybe that will help you get the right answer.
Is it option C, phenylalanine ammonia lyase PAL, cinnamate 4 hydroxylase C4H, and then 4Cl?
Right. So, what is the substrate and the product for PAL? Any idea?
The 4-coumarit.
No, no. What's the substrate for PAL? We'll go with that. You can just
Four,
list out
first one is
for
para.
phenylalanine ammonylase. What's the substrate?
You're asking phenylalanine, then after that, that ammonia lyase, we will get tranexamic acid.
Huh. Okay. Now tranexamic acid is there. That is the substrate for which enzyme?
Phenylalanine ammonia lyase.
No, no. So substrate is the one that is getting converted. Product is the one that is being formed. So for phenylalanine ammonylase, phenylalanine is the substrate and transinamate is your product. For cinnamate 4 hydroxylase, transinamate is your substrate. What is the product that you get?
For the C4H, cinnamate 4 hydroxylase.
Yes.
Uh, uh, that is 4-coumarit we will get.
4 coumarin. Okay. And what is the substrate and product for 4 hydroxycinnamoyl CoA ligase? for 4CL?
CL, it will be for combined after that 4 hydroxycinnamide is it caffenyl? Wait, I'm not sure.
4CL, it adds,
Paracoumaryl coel,
yeah,
coel,
CoA, yeah.
flavonoids, cyanine, tannins, yeah, tannins. Okay.
So this paracomaric acid is can also be
Paracoumaryl
called as 4-coumarate. So 4CL is 4-coumarate CoA ligase. So it's a ligase. It covalently
ligase.
attaches this CoA to this position. That is what is happening chemically.
So here I have a question for the pulp and paper making, the populous tremule sites. In this, what happened there? Outcome is the significant reduction in lignin content. So in this particular, the repression of lignin biosynthesis promotes cellulose accumulation. In that particular, I have not understood the thing in the class, in the
I
videos.
didn't I didn't understand. Is this related to that question?
This
Then
is the question. Yes.
okay, right. So what they say is here in an attempt to reprogram lignin biosynthesis pathway in Populus tremuloides, scientists have introduced antisense RNA for 4-hydroxyl cinnamoyl CoA ligase, which is 4CL. What was the outcome of this experiment observed in transgenic trees? And they,
So here it is
huh,
that 4CL, no, sir. But
yes,
in the case of the another thing was that overexpression of the CALT SH. So in that, what happens, sir?
can you repeat overexpression of?
The PT CALT SH.
Can you type it? It's not clear to me. Can you type it in the? You can type it.
PCALDSH.
You can type it here. I will just look at the full form of the enzyme, and then once you type it, I'll just know what enzyme you're talking about. Yeah, Swati, can you type it? The enzyme.
1 minute, sir. I'll type.
Yeah. Sure. PT Cald SH. Okay. PT Caldi. So I haven't come across this. Let me check. PT could be the name of an enzyme. I'm not sure. I'm not able to get that. Uh, where is this question posted?
It was there, I think, in the
No,
write it
is
down like
this
that.
PT
Assignment.
Cal? See, shRNA is short hairpin RNA. Again, this will also remove or reduce the protein level. So the enzymes expression levels will be reduced. So accordingly, you need to check where this PT Cal is being used. I am not able to recall at the moment what this PT Cal is. And the next question is, I think it's constitutive and not consecutive. Just tell me if I'm wrong. Constitutive promoter. So this is cauliflower mosaic virus, I think. CAMD35, which is a strong promoter.
Sir, that enzyme that is it called 5H?
It, I don't think, see, SH is short hairpin. So in RNA, you can form secondary and tertiary structures that would prevent translation. So I think this is similar to what we saw earlier. I'm not sure about the
No, the
enzyme
question
alone.
was, yeah, the question was asked that what happened to the lignin significant reduction or increase in S-lignin? So there is a confusion in the that particularly that the CALD enzyme, that overexpression. That's why I asked.
I'm not able to recall what this PT cal is. That's the. Let me check again. But all I know is if you put this shRNA, you will get reduction in the enzyme level. Are you sure it is PT cal? Because uh, I haven't come across this. Like C4Cl, F3HFL, less, all of these are familiar to me, but I have not come across this enzyme. Maybe the name is given wrong. Were you able to post this message on the platform, that Google Groups?
Okay, so this is the PT CALD 5H, sir. The question I checked.
Okay,
PT CALD 5H in
PT
that
cal
particular
5.
plant. Yeah, that Populus uh, tremu tremulcite,
Tremuloides. Okay.
tremuloides. Yes, PT CALD 5H
Mm,
overexpression. What happens in the S-lignin increase or significant decrease in the this one? Um, that has been reduction in the lignin.
hmm.
And another said the correct sources of H-lignin, is it angiosperms?
Uh, have you submitted the answers to this assignment?
Submitted, but we can no, no, not like that. I'm
Oh,
just asking that because it was not mentioned that correct source of H lignin, like angiosperm somewhere means it is not mentioned. That's why.
so you need to know where the enzymes for the lignin
Even that
pathway.
yeah, constitutive that was already submitted last week.
Okay,
So that also I was not aware like what to that was wrong. So that CA you told no, sir, that is the correct one.
okay, okay. So this PT-Cal, because you're telling lignin, I think it could be coumaryl aldehyde. Based on the name, I'm telling para coumaryl aldehyde, something of that sort. It should be that way. And so, if we look at the types of lignin, like we saw earlier, G lignin, S lignin, and H lignin, let me see. So, we have 4-hydroxyphenyl, guaiacyl, and syringyl. We need to see which of these could possibly have paracoumaryl aldehyde. Syringyl, SYLNGyl, syringyl lignin. Yeah, you're able to see that. Paracoumaryl aldehyde. So we can see biosynthesis pathway for all the different lignans. We need to identify where this CALD is. LD5H is 5-hydroxylase, aldehyde 5-hydroxylase.
Sir, the last question we were discussing, no, sir.
Yeah, this 1 minute. I'll go to the. Okay. Huh. Yeah, again, here they ask about what happens when you have an antisense RNA for 4CL. So where exactly does 4CL act? So this is the paper that has been used for framing this question, and what they mention is when this 4CL is downregulated, it reduces the lignin content in the cell wall. So they used this strategy and they tried to correlate it with 4CL RNA expression levels. And they observed that small lignin reductions of about 10% were associated with reduced productivity and decreased wood syringyl or guar acyl lignin monomer ratio, which is G and S lignin ratio, and small increase in H lignin. So there is increase in H lignin and decrease in S and G lignin. This is what they observe. So what they ask in the question is significant reduction in increase in lignin, reduction in cellulose, increase in both, decrease in both, and then vice versa. So what happens to lignin and what happens to cellulose is the question. What we observe from this paper is that there is decrease in lignin. They also mention here that there was no evidence that substantially reduced lignin contents increased growth rates or saccharification potential. This is because they were able to see increased cellulose content, but they couldn't correlate that because of reduction of lignin content, there is increase of uh, cellulose content. They are not able to tell that this is the reason, but they observe both of these two trends. That's what they mention here. So it is option D for this question. Uh, was it clear?
Okay, sir.
Right. Right. And yeah, I with this we come to an end of this week's session. If you have any more questions, you can ask me. I'll be on the call.
Okay, sir. Thank you, sir.
Thank you.